Welcome, everyone. We are coming on the end of week 2. We have two chapters, chapter 4, prokaryotes, and chapter 5, eukaryotes. Um, we're going to do a pre-assessment, quick little Quizlet, just to get us warmed up. We'll go through our live session. If we have time for Kahoots, we'll do it. If not, no worries. I've already placed it. It'll go in the archive, so you guys will have it as an additional resource, as well as this pre-assessment. So you guys will have both for your upcoming final and to kind of use as a resource for this week. Now, keep in mind that the highly missed questions from week 1, I'm working on them, and you'll see them tomorrow in announcements, just if you want to kind of look and check and see. And I usually do that every Thursday. So next Thursday, you'll have week 2 and so on and so on. And you can always use those as, again, additional resources as you guys are gearing up for not just this quiz, not just your midterm, but even your cumulative exam, because it's going to be here before you know it. So right off the bat, we'll jump into a little Quizlet. Okay. It's not long. For those of you who cannot join, let me put the link in. I don't know if this is gonna work. Let me see if I can put the link in here. And of course, there is the um, QR code as well. And I turn the music down because it's very loud. See if I can grab another one. And that way, those who are watching will be able to see the same questions. Let me go to settings really quick because this is going to be really fast. Fast mode, only 12 questions, 3 winners. Perfect. Okay. So there are 9, there are 15 of us here. Let me share my screen. Okay, so for those of you who are not able to join, or if you're still watching the archive, you'll see some of the same questions on the left. And for those who aren't able to log in with me tonight, you can kind of put your best answer into the chat and I will fill that in for you. So let's go ahead and start the game or the pre-assessment. All right, here we go. There we go. And again, there's only 12 questions, okay? So it's right here. Let me type it in there for you really quick. log in, you can kind of see on the left some of the questions that they'll see or that they're seeing. And if you want, you can type in what you think it is, and I'll mark it for you as well. We have one. I'm waiting for two more. Keep it up, guys. Good job, everybody. Good job. Okay, so this was just a pre to kind of get us, get our brains ready, warmed up as we go through the course tonight. Okay, now let me make sure I share this one too, because I want you guys to have both your Quizlet and the Kahoot. Okay. All right, so that was our little warm up. So we're starting off with prokaryotes. Okay. Now with prokaryotes, we've got to talk about some stuff. We're going to talk about terms. We're going to talk about the difference between archaea and bacteria. We're going to recognize bacterial shapes, how they compare, understand what prokaryotic cell parts are even all about, explain bacteria, how they're grouped. We're going to understand why bacteria make spores. Now, this is really important because this information we're talking about now is what we're going to be talking about in the beginning of theory 2 when we get into sterilization and disinfection. We also have to connect everything on why it's important to us in surgery. Now, I want you to know that microbes are everywhere. Okay, they're on us, they're in us, they're all around us. So we get into terms like sterilization, all right? We're talking about killing all microbes on surgical instruments because we want to prevent infection in deep tissues. Remember, we're sometimes doing big belly cases where the patient has an incision from midline all the way down to their pelvis. Okay. And we're literally going through and checking their intestines, making sure there's no blockage or different things. With sterile technique, this keeps our field. The surgical field is the patient. Okay, when we talk about our sterile field or surgical field, that is our patient. We want to keep everything clean and free from contamination. Now, with aseptic technique, this is reducing germs, but it doesn't make things completely sterile. So I want you to think about when we cleanse our skin before surgery, we are including scrubbing, chemicals, because we're trying to just slow down microbial growth. You may see a concept asking, can skin be completely sterile? The answer is no. Skin cannot be completely sterile. So microbes can still cause infection. Now, we try to prevent infection by following strict procedure and using proper skills. So we look at archaea. Archaea are different from both bacteria and eukaryotes. Remember all those systems and phylums and stuff we learned last week? Now we're going to break them down. Archaea include things like methogens. These are microbes that produce methane gas and live without oxygen. There's halophils. These are those salt-loving microbes that thrive in oxygen-rich environments. There's also thermocytophiles. These are heat and acid-loving microbes that live in hot, acidic places. Okay, we got to break down the basics. All right. Bacteria are classified into different groups, and it's going to be based on specific traits. Morphology, for example, shape, structure. Staining is how they react to stains, gram positive, gram negative. We're going to break this down in this course. But we also need to know how they grow. What condition? Anaerobic, aerobic, facultative, both. How they move is another one. We look at the type of proteins they produce, their DNA structure, sequence, and what they actually need to grow. And we can't forget their pathogenicity, their ability to hurt us and cause disease. Now, with morphology, we're talking about the basics, okay? They're classified by their shape, their arrangement. Round, pairs, single, groups of 4, cubic packets of 8. They could have staphylo, which means clusters. So we talk about Staphylococcus aureus a lot in this course. Staphylococcus pyogenes a lot in this course. So we're looking at clusters. We look at bacillus, rod-shaped, mono, single, diplio, pairs. Now, staining itself, looking at the middle, is so important because we want to determine the pathogenicity and antibiotic resistance. There are conditions, right, necessary for growth. How viable are they? Bacteria grow by binary fission, where one cell divides into 2, and that increases the population, not the size. Now, colonies are a group of bacteria, each originating from just one cell. So we got to look at the anatomy of a prokaryote. It's a simple cell without a nucleus. You will see concepts again. It has the following parts, though. Has a cell wall, gives it shape, protects it, kind of like our skeletal system, okay? Has a cell membrane. It controls what goes in and out and leaves the cell. Your cytoplasm, if you look, your cytoplasm is jelly-like substance. It's inside the cell where chemical reactions actually happen. DNA, it's a single loop. It's all the genetic material. Ribosomes, these are going to be the tiny structures that make up proteins. Your flagella, they are the tails, okay? It helps with movement. And then the pila, look at these smaller hairs. What they do is they attach to the cell, okay? Kind of like Spider-Man has his web. These pila are like the webs that attach to whatever it needs. Now, prokaryotes are much smaller and simpler than eukaryotes, and that's going to be really important because you're going to have to differentiate the two. Eukaryotes are more like human cells because they have a nucleus. It's really important. Now, most periodic cells have a rigid cell wall that surrounds the plasma membrane. And again, that gives it its shape. In eukaryotes, vertebrae, like humans, we don't have a cell wall, but plants do. So we look at the cytoplasm or the plasma membrane. In both prokaryotic and eukaryotic cells, the plasma membrane is a flexible layer of phospholipids with proteins that are embedded in it. And what that does is it controls what enters and leaves the cells, like a door. The door is open, things can go in and out. If it's closed, it can't. And then we get our capsule down at the bottom. Now, many prokaryotic cells have a capsule, which is a sticky outer layer made of sugar polymers. Now, the capsule helps prokaryotes also stick to surfaces. And think about it, like sugar, if it gets wet, it kind of gets sticky, right? And just the same, it helps it to cling to each other, and it also acts as a protection for them drying out. Now, what is a capsule and why does it matter? Again, that capsule is thick. It's a protective coating. It's made of sugar or proteins. It's surrounding some bacteria. I want you to think of it as a stealth shield. It helps bacteria not only hide, but it protects them from the immune system, like our immune system. In the operating room, knowing which organisms have capsules matter because they're harder to kill and more likely to cause infection, especially if sterility is broken. The chapter talks about immune evasion. Normally, immune cells like neutrophils and macrophages, they detect, they're like, okay, something's wrong. There's bacteria in here, and they can recognize antigens, surface markers. But these strong capsules cover those antigens. So now bacteria are almost invisible to our own immune systems. Even when detected, capsules make it harder for immune cells to kind of grab onto them and engulf the bacteria. This process is called phagocytosis. Now, what happens is the infection sticks around longer and is harder to clear, which can be very dangerous, especially after surgery. One of the examples the chapter talks about is Streptococcus pneumonia or pneumonia. This is a common bacteria that causes pneumonia meningitis and, unfortunately, SSIs, surgical site infections. If a capsule is its main virulence factor, without it, the immune system could easily clear it, like knock it out. This is why capsule-based vaccines like pneumococcal vaccine work because they train the immune system to kind of recognize, hey, that's not normal. That's a capsule. It's not invisible. Go get it. Does that make sense? So I want to make sure I'm breaking them down. Okay. Now we get into the cytoplasm. Cytoplasm refers to everything inside of the plasma membrane, including the fluid and the cell parts. Chromosomes in prokaryotes, like E. coli, have one single circular DNA that contain all of their genetic information. Some prokaryotes, however, can have up to 4 chromosomes, which may be linear or even circular. So it could be kind of like in a line or in a circle. So what are we looking at? So again, when we break it down even more and we look at the flagella, okay, you see these little Medusa kind of things going on. They are found in both prokaryotes and eukaryotes. Again, the pili, short, hair-like structures. Now, they do help with movement, but their main job, again, is that being able to stick onto different surfaces, which can lead to infection and is an important factor in how harmful bacteria can be. And when we talk about spore formation, because it's one of the biggest things we're trying to kill in decontamination and sterilization. Now, these endospores are tough. They're dormant structures that some bacteria, mostly gram positive, form when nutrients are scarce. This whole process helps the bacteria survive in harsh environments. Now, again, the flagella, tail-like structure, that's what's helping it move. Movement is so important for bacteria to be able to invade tissue and for human cells like sperm to function. When we talk about prokaryotic flagella or bacteria, I want you to think of it as a tiny outboard motor. So you're on a boat and you know they have the motor on the outside. It moves like propeller like rotations. So it'll spin clockwise or even counterclockwise. Okay. It's powered by a proton motive force. So that's energy from movement of protons across the cell membrane. This type of movement helps the bacteria swim toward nutrients or away from danger. When we talk about eukaryotic flagella, you're thinking of sperm made up of microtubules in 9+2 arrangement, which is important for lab exams. And it moves with more of a whip-like motion. So it's not spinning. So it's actually moving like this versus the prokaryotic is like this. Okay. So making sure you're able to distinguish between the two. We also look at ribosomes. Okay. Protein factors. Ribosomes make proteins, which bacteria need to grow and survive. Ribosomes are like protein factories. They build proteins that cells need to actually survive. Each ribosome is made up of two parts called subunits, like two pieces that really fit together to do their job. For bacteria, we look at prokaryotes. You have small pieces like a 30S or big pieces like 50S. Okay. For humans, when we look at eukaryotes, they're smaller, 40 and 60 for the big. Now, S, because you see the S on the bottom of these, right? 70S, 80S. This stands for Sevberg units, which measures how fast the pieces sink in centrifuge. Not size, but it helps scientists describe them. So it's a scientific symbol to help them know what it is. Okay. But that way you can kind of understand what that S is. Like, what is the S? All right. Now, with certain target bacterial ribosomes only, we want to know how we can kill it. So we look at what's called selective toxicity. We're trying to kill the bacteria and also leave our human cells unharmed, which is really important. So, like, let's say a bacteria attacked to an 80S ribosome. Now, keep in mind, eukaryotic ribosomes are in humans. So, if it's an 80S and it kills it, it could also harm cells, making it toxic for us or our patients. A couple examples: tetracycline, okay? No protein synthesis, bacteria die. We have erythromycin. It stops proteins. But why is this important for us in surgery? Prophylactic antibiotics is what we kind of do before to prevent, is given before surgery, like cefalozin. And it's chosen because what it does is it targets the bacteria without harming our patient's cells. So if they didn't understand that, or we didn't understand that, it could lead to wrong antibiotic choice. We could grab something that would actually harm an ADS eukaryotic ribosome, which could actually harm our patient. So for us, this is why it's so important for us to kind of just hone in on surgical technique and proper antibiotic timing. Luckily, our anesthesiologists are usually the ones giving that prophylactic antibiotics, but it's still good to kind of know, okay, they've got it on board, meaning it's in their system. Now, we're going to talk a lot about biological indicators in Module 3, but it's really important for you to understand that this is the golden standard. This is how we determine if our instruments are indeed sterile. It tells us if the condition needed to kill a certain number of microorganisms were met in the sterilization process. Now, BIs are going to be done weekly on each sterilizer to just make sure that the sterilization process is working properly. Okay. Now, that was prokaryotes. Not too long, right? Now, when we look at eukaryotes, we are going to still be looking at terms. We still got to tell the difference. We're still going to be comparing prokaryotic cells and eukaryotic cells, but we're going to break down that eukaryotic cell just like we did the prokaryotic cell. We're going to explain how microbes really interact with their functions, compare the three types, hint, hint, of eukaryotic microbes, and connect it to why it matters to us in the operating room. So I do have some images, okay? First image you see in front of you, we're talking about fresh tissue specimens. Fresh meaning no fixative, no formalin, because we want it not to be ruined when it goes to the lab for testing. So examples, most likely what you see in front of you is some type of tissue. Could be breast tissue, could be from the lung, it could be from anything, okay? It has to stay fresh for the hormone receptor testing. So keep in mind that if you put like formalin or another fixative, it can actually kill human cells, making tests for treatment planning inaccurate. If you're in a case where we're doing staging, we're going to be taking a lot of different portions of different cells, and you'll have a runner that once we grab this, we take the specimen from our surgeon, we send it to the runner, and then we're waiting for pathology to read that in real time. Staging cases can be anywhere from an hour to longer, depending on if the margins have been removed properly. The one thing I want you to remember, and we'll go over it again next module, is we always confirm with our surgeon how the specimen should be handled before sending it to the lab, because mistakes cannot be undone. All right, just trying to go fast. Now we get into the cellular empire, prokaryotes versus eukaryotes. Remember, prokaryotes are simple. Um, to visualize, I want you to think of a human hair. Okay, it's about 70 thick. Most bacteria are about 1 to 10 long. Human cells are usually 10 to 100 in size. Is just 1 micrometer, equaling 1 millionth of a meter. Okay, just in case any math prodigies are in here. When it comes to eukaryotes, we're talking about more complex. They're going to be larger. They have a nucleus and organelles. They include protozoa, algae, and fungi, and even humans. Now, knowing the difference helps us understand how microbes cause infection and why sterilization is so important. Again, it has a plasma membrane, protects the cell, maintains a stable environment inside the cell. Several important functions: transporting nutrients into the cell, removing toxic substance from the cell. Cell walls in plant cells, remember, the cell wall surrounds the plasma membrane. Um, it helps with tensile strength, resistance, strengthening or stretching. It also helps with protection against mechanical and osmotic stress. Changes in pressure, okay? The nucleus controls and regulates the cell's activity, like growth and metabolism. It also carries the genes which contain the hereditary information. Now, we have to talk about how species interact. First one you see on the left is symbiosis. Close relationship between the two species where at least one of them is benefiting. So you have different types. Mutualism, both are benefiting, right? And communalism, one benefits, the other one's like, whatever, I'm fine. So what you see is a remora fish, um, and it's riding the back of the sharks. It gets food scraps, and the shark isn't harmed at all. It's just, okay, they're floating with me, that's fine. You also have parasitism, okay? One benefit, one is harmed. Fleas on dogs. The flea is getting their buffet on, and the dog is suffering. It's like, okay. But why is this important to us in the operating room? Now, microbes can have similar relationships with humans. Some help us, some don't. No one is going to really help with infection control and patient safety. Now, can you guys think of a situation that could happen in the operating room where a normal, harmless microbe might actually become harmful? And I'm going to give you a scenario. Is it possible that if the skin barrier is broken during surgery, keep in mind, we're giving the surgeon a scalpel for them to cut the skin. Normal skin bacteria, do you think it can enter deeper tissues and actually cause infection? Yes or no?
Yes.
Yes, most definitely. You guys are correct. And again, we have normal flora on our skin surfaces, and the surface is not doing anything, not harming us. But if it gets into our deep tissues in an area or an environment that it's not known to be, it can definitely cause an infection to our patients. Fungi are eukaryotic, like us, believe it or not. So they have a nucleus and organelles. Now, this is important because drugs that kill fungi can also damage human cells because they share so many similarities to what we normally have. Now, both fungi and human cells have what's called the cell membranes, right? Organelles, and we also have similar metabolic process. This why it makes it so hard to create antifungal drugs that target only the fungus without harming the patient. Now, antibiotics, on the other hand, work well because bacterial cells are completely different from ours. So it's easy to be like, oh, this is bacteria. Let's give it a bactericidal if we're going to clean a different surface. Now, most antifungal drugs target a component in fungal cell membrane. Keep in mind, human cells have cholesterol instead. So this means that there's a narrow target, limited choices for antifungal medications. Some drugs can actually cause damage to the patient's liver or kidney. So, a lot of times when we have patients that are on a lot of antifungal medications, they're getting liver and kidney tests just to check the function to make sure that it's not being destroyed or affected adversely. Candida infections are a big concern in immunocompromised patients. Okay. Um, this is when sterility is broken during surgery. So, here's an example. If a central line or a surgical wound gets contaminated with candida, the infection can spread quickly. And keep in mind, it's a fungus, so it makes it really hard to treat. And this is another reason why we are so strict on aseptic technique. Um, Really, because prevention is a lot easier than treatment. Yes, like toenail fungus meds can cause liver issues, most definitely. Now, I keep talking about candida. What is it? It's actually a yeast. It's a type of fungus. It normally lives in the human body. So it's normally there. Our mouth, gut, vagina doesn't cause any harm, but becomes a problem when the immune system is weakened or barriers are broken, like after surgery. If you do a lot of oral maxillofacial cases, we're very, very strict to make sure that like everything is irrigated properly. Okay, because you don't want an infection. Because you guys are going to learn in theory, I think in theory two, how your sinuses and everything is so connected that you have to be careful. There is also an instance where if a patient comes in and they're disoriented, ENT and neuro will be called because we want to make sure that if it is pus, it's either in the sinus cavity, not in the brain, and vice versa. So we also look at endosymbiotic theory. Okay, about 1.5 million years ago, some prokaryotic cells joined forces and formed a mutual relationship, and it created the first eukaryotic cell. So there's some evidence. The chapter talks about mitochondria and chloroplasts having DNA. Their DNA looks a lot like bacterial DNA, not actually human DNA. So ribosomes are bacterial-like. These organelles make proteins that bacteria do. Now, chloroplasts have chlorophyll, same pigment as photosynthetic bacteria. Now, it shows how life evolved and why some parts of our cells act really like tiny bacteria inside of us. Protozoa. And of course, I'm an image person. I'm the type that I have to see it. Okay. Now, we look at single celled organisms. They can live freely as parasites, they feed on organic matter. Okay. Algae, they live in the water. They make their own food and they use sunlight, which is really important. So examples would be like seaweed, pond scum, algae blooms that are in lakes. Now with photosynthesis, think back to where some plants or processes plants and some algae used to make food. So they need sunlight plus water plus carbon dioxide to make sugar and release oxygen, which is important for life. Now, why should we, as surgical techs, care about organisms like protozoa or algae if they don't usually live in the operating room? What do you guys think? Okay, I'll give you a hint, or I'll tell you. Now, the reason is some protozoa cause infections if sterile water or instruments are contaminated. Algae-based contamination in sterile solutions or systems could actually cause or create that infection risk. So this is why we really look at our water quality and sterilization, making sure that the machine is working properly. I did see a question. I want to make sure. Why does it grow with antibiotics? Now, that is a great question. When you ask why does it grow with antibiotics, we're usually observing the bacterial growth itself or in the presence of a drug that's designed to slow it down or actually kill it. Something that seems like nothing big at first glance. Now, we think about how it happens. The most common reason is that microorganisms have unfortunately developed resistant mechanisms that allow them to survive and multiply despite the presence of an antibiotic. So it's almost like even though you're throwing different things at it, it has a shield protecting it from it. Some examples would be we use enzymatic destruction of a drug, where we try to get the lactate out. We try to destroy, um, it'll destroy like penicillin and any type of related antibiotic. So the bacteria becomes resistant to that. Now, when the antibiotic binds, the drug can no longer act or attach. So it's almost not only a shield, but it's a counterattack. Okay. The bacterial cell also can pump out the antibiotic. So it's like it'll let the antibiotic come in and push it out. So it didn't even do what it was supposed to do in the first place. And that also prevents it from reaching different toxic levels that are inside. So it's still growing. Does that make sense? Kind of like to break it down a little bit more. Okay. Fungi. Now, fungi is plural of fungus, believe it or not. It's a group of organisms that produce spores and feed off organic material. Some fungi cause serious infections in our patients, especially if the patient, again, is immunocompromised. Okay. Spores can survive in extremely harsh conditions. So, this is another reason why in the beginning of Module 3, we stress sterilization and we stress it in Module 2 in micro as well. So they can be found in a lot of different forms. Molds can be in the air. Yeast can get into the bloodstream. Also, mushrooms and toadstools. Not much that you would see, but it's still good to know that they're there. So here's an example. A patient in the ICU has a major surgery. It develops candida infection through a central line. Again, we know that treatment is going to be difficult because antifungal drugs are limited and often can become toxic. So then we go back, and this lets us know that we are reinforcing strict aseptic and sterile technique when the line is being placed. Okay. Again, I should have gave you a forewarning images. Um, there are key facts for surgical techs for us to kind of know. Okay, fungi, they're eukaryotic organisms, meaning they're complex cells, found as molds, mildews, yeast. About 400 species infect us, humans. They're not plants. They can't make their own food. They have to be fed on organic matter. They grow well in dark, moist areas. They use enzymes to break down material or nutrients. Now, yeast, think of bread, beer, wine. Mold, think of our source for penicillin, the first antibiotic. Molds have filaments that form mycelium. It actually reproduced by spores or tiny, tiny seeds. Now, fungal spores can survive in the environment and cause serious infections. This is why we go back to that BI, the little small biological indicator, and we use that to make sure that The machines that we're using to sterilize our instruments are able to kill the spores. If sterility is compromised, our patients can unfortunately get an infection. Okay. Now, the last part we're going to talk about before we get into our Kahoot is the nuclear envelope. Surrounds and protects the DNA. It regulates transport in and out through the nuclear pores. You guys can kind of see the pores here, has a hole. These are like the doors, okay? Prokaryotic cells lack this membrane compartment. So they don't even have it. That's why they're in the eukaryotics. All right. Now, not a lot in these two chapters. Again, you have your Kahoot, and we're going to do this Kahoot really quick, and then we're going to do a little quick class activity. Let's go ahead and see what you guys know. So what I do for Kahoots, just so you guys know, I try to grab the two highly chosen questions. One's correct, one's the one that's highly chosen. And I give you 10 seconds to answer the question. Now, I know at first you're gonna be like, oh my gosh, this is so fast, but trust the process. What happens is a lot of times we second guess, like we know the information, but we're like, maybe, I don't know. I'm really bad at second guessing. So what this does is it doesn't give you that much time to second guess, but to really trust your instincts, okay? Is there a link? Let me see if I can put this in. I'm not sure if this is going to work. See if that works. And then the pin is 5102694. For a fact, let me click on it and see if it works. If I get another, it lets me do it. Okay, it'll let you do it. It should let you do it. Now, for those of you who cannot log in for whatever reason, it's okay. You'll be able to kind of pick red, 1, 2, once the game starts. So there's 17. I usually kind of start around 11 or 12. So we're at 10. For everyone who can't join, remember you can still put in your answer in the chat, okay? You can still put your answer in the chat, and that really helps with your participation as well. So let's start. There you go. What shape do bacteria call coccus? Okay, so 10 seconds, guys. Round. Good job. Number 2. Which term refers to bacteria arranged in clusters? Clusters. I kind of mentioned that we will be talking a lot about some of these. That's right, Staphylo. So Staphylococcus. Specifically Staphylococcus aureus, we talk a lot about in this micro course. Which term describes bacteria in pairs? In pairs. Yes, diplio. Go with your first instincts. You guys know this, I promise you. Question 4. What is the term for bacteria that are arranged in groups of 4? Groups of 4. This one is tetra. Think of the game Tetris. I don't know, I think of that one. I was like, oh, Tetris. What does sarcinae refer to in bacterial morphology? I thought I only had 2. I'll have to go back, you guys. I kept all 4. Sorry about that. I thought I switched it to 2. Cubic packets of 8. Okay, cubic packets of 8. I have seen that concept. What does mono refer to in bacterial morphology? Mono. Single cell bacteria. I always think of mononucleosis. What shape do bacillus bacteria have? Rod shape. So sometimes I try to throw pictures in to kind of connect the two. So rod shaped. Good job. What shape do Vibrio bacteria have? Kind of have to look a little bit close because these are comma shaped. What type of bacteria arrangement does palisade describe? These are fence-like arrangements. And the last one. Which of the following describes bacteria in a chain arrangement? Chain. Process of elimination. Strepto. So remember, we've used Staphylo, we used mono, and we used tetra. Sometimes the quizzes, especially even on the CST, will kind of throw the different concepts, but will throw a different answer with that same kind of concept frame. And let me make sure you guys have this link as well. Again, for those of you who are watching, you'll have both in the live archive section. All right, good job. We just have one final little thing that we're gonna do before we get out of here. All right, I want you to take a good look at this picture, okay? Good look. Get it into your brain cells. Everything that you see from protozoa to rough ER to phagocyte, okay? Guys good? All right. So now that you've gotten a good look at it, you're gonna have a couple minutes, okay? These are the terms that you're gonna need. These are the same terms we just saw. I went too fast. These are the terms you just saw: protozoa, mutualism, communalism, rough ER, phagocyte, Golgi, fungi, parasitism, and algae. Now, again, this is on the honor system, okay? We're gonna look at the pictures, and I want you to see how many you can identify. Okay, I'm gonna give you a couple minutes to see what you can identify. Ready? Okay. Well, 30 seconds, kind of see which one in your mind, visualize. Do you know which one is which? And then let me know when you guys are ready for the reveal. Okay, I see you're ready. All right. And again, this is the honor system. I literally put A, B, C, and D. So we're going to go back. Okay, here we go. How'd you guys do? Pretty good? So-so? So-so. Okay, That's fine. That's why we're here. All right. Not bad though, because remember, you guys just saw this just now. All right. What questions do you have for me? That's pretty good. No questions yet? Okay. Well, with that, for those of you who are watching the archive, I'm curious, how can knowing the difference between prokaryotic and eukaryotic cells guide your choices for sterilization in the operating room? Why does understanding bacterial spore formation matter for preventing infection during surgery? And how can knowledge of microbial relationship help reduce infection risks in the OR? We talked a lot about prokaryotic cells, broke them down from what's in them to the importance of spore formation, and eukaryotic cells, which are more complex, just like fungi, protozoa, and algae. We talked about their structures, how they interact with us as humans, the relationships with mutualism, commensalism, and parasitism. We also talked about why it's important to understand in the operating room. Now we're at week 2 and it's going to go by so fast. And what I mean by that is in 3 weeks is the midterm. We're going to be ready. So keep in mind that tomorrow you will see the highly missed questions from week 1. If there is something that stood out, or if you notice that maybe you looked in the book and something was marked differently, y'all let me know so that I can fix it for y'all as soon as possible. With that, I do want you guys to have a wonderful rest of your evening. I know this was a lot of information, so you may need to walk around, take a breather, be safe this weekend, and I do look forward to seeing everyone who can attend next week's live session. Good night, everybody.